Thank you so much for joining the webinar today. I am Vidya Sundaram. I am the VP of BizDev at BioChain. I will be moderating today's webinar titled Decoding the Complexity of Life, a Journey into Spatial Biology Services. So BioChain has been established from 1994, and we have been a leading biopharma, biosample specimen and analysis company. And we've been providing these services to biotech and biopharma from 1994. Since 21, BioChain has also been a provider of spatial biology services. We are in a very unique position because we provide multiple spatial biology technologies or platforms. We provide, for example, 10x Visium CytAssist, NanoString's GeoMx Digital Spatial Profiler, CurioBio's SlideSeq, and StormX StereoSeq technologies. We've also partnered with Enable Medicine for Akoya Codex and spatial biology data analysis services. So BioChain is a one-stop shop from sample preparation to data analysis for all spatial biology projects. We will be having a series of webinars highlighting each of these unique technologies in the next few months. So stay tuned for announcements from our team. And in today's webinar specifically, we will be highlighting different spatial technologies that BioChain provides. 10x Visium, CytAssist technology, which is unbiased, where you can take the whole slide and look at the WTA, whole transcriptomics, and GeoMx, which is got a very unique technology where you can actually select regions of interest, cell types that you're interested, and look at the whole genome expression. So we will start off with a pre-recorded presentation by Dr. Rikita Gadkar, who will discuss both the spatial biology technologies. We will also have a live Q&A session at the end of the presentation with our spatial biology technical team lead, Dr. Elim Cheng. So if you have any questions in terms of projects, spatial projects design, please feel free to chime in the Q&A box. So let's start with the pre-recorded presentation, and we will all be here if you have any questions. Thank you.
Thank you. Thank you, Vidya. Hi, everyone. So thank you, Vidya. Thank you, everyone. Thank you all for joining today. My name is Rikita Gadkar, and I'm very happy to have this opportunity to walk you through the journey of how we started exploring spatial biology and where we are right now and where we expect to go. And we started this journey back in 2021 with 10x Visium and GeoMx platform from NanoString. Since then, we have grown along the way, overcoming the many road bumps that we came across and learning from them and expanding further to provide different spatial services all under one platform, all under one roof. So to begin with, I would first like to give you a brief introduction of what BioChain is, who we are. So BioChain has been engaged in providing biological samples since 1994. That's for the last 29 years. And we have been accelerating disease diagnosis and drug discovery research with complete solutions from sample collection to preparation and analysis. And our aim is to provide quality and innovative solutions. And we understand that no one solution fits all problems in science. So we are working along with you to custom design solutions to answer the specific questions that you have on hand. So the products and services offer under these 6 main categories. We offer biospecimens, which include frozen and FFPE tissues. These include sections, blocks, panels, and arrays. and nucleic acids and proteins. We also provide matched samples and custom procurement services as well. Our automated solutions include kits and instruments to make your day-to-day life easier, more specifically AnaPrep instruments and related kits. And we also offer reference and control standards, which include FDA arrays, nucleic acids, and proteins. Our most famous reference products are our Cyno-Monkey and Rhesus Monkey products, besides others. And we offer OEM services for large life technology clients and also offer extraction and purification kits as well. One of our hot selling product right now is cell-free DNA extraction kit, which has been proven for better yields amongst the competitors. So as I mentioned before, back in 2021, we took our first step in the spatial biology services and became certified service provider for the 10x Visium digital spatial gene expression service, as well as NanoString GeoMx digital spatial profiler. And we have since continued to expand our network of partners and collaboratives in this field, and we hope to continue to do that. And so with that, for the sake of this presentation, I will expand further our experiences into the spatial biology services. And so as we began back in 2021, we quickly realized we can be an end-to-end service provider, with our in-house tissue repository, our pathologist, and scientific team. And we started with a few simple projects using our in-house tissue and TMAs to learn the steps that were involved in these workflows, the sample preparation and optimization and the entire workflow for both 10x Visium as well as NanoString GeoMx platform, all the way up to reporting and understanding the data. So before I head into some case studies, I want to briefly talk about our tissue procurement. And we have a repository of retrospective collections, which includes frozen and FFPE tissue. These are IRB approved and donor consented samples selected by our board certified pathologists. We provide match sets with tumor and normal adjacent tissue from the same donor and cancer cell lines and the clinical data associated with them. Also, we provide primary and metastasis samples, plus some rare disease tissues such as Alzheimer's, Parkinson's, glioblastoma. And besides our repository and prospective collections, we also work with the customer provided material. And, as an example, I'm sorry, give me a second here. I just want to make sure I cover everything and don't derail from the topic. It's just so easy to do that. Okay. So our first few experiments, we used some lung, brain, breast, and colorectal cancer and normal tissues from our repository for 10x Visium study and a breast cancer TMA for GeoMx study. I will talk about the results from these studies in a few slides, I promise. But first, let's take a look at sample preparation and optimization, which is the next step once you have your, tissue. So let's first talk about sample preparation for 10x Visium. Frozen tissue, 10x Visium sample preparation can look very different depending on the type of tissue. So like frozen tissue with direct placement requires OCD embedding and tissue optimization. Tissue optimization, as Samantha just was explaining, it can be difficult even for a histopathologist, experienced histopathologist. Tissue optimization and slide preparation is a very tedious and time-consuming process. So in the beginning, it seems like FFPE, FreshCut was a more quicker option as far as the workflow was concerned. Also, with direct placement, rinse score or DV200 qualification, which is much higher, 7 for rinse score and 50% for DV200. This was a limitation for some of our customers who were working with some rare and difficult tissues. So fortunately, these issues were addressed with CytAssist, and as Vidya mentioned, we took a leap and bought the CytAssist, and which does not require tissue optimization for frozen samples and can take RIN score as low as 4 or DV200 as low as 30%. In addition, we can also use archival FFPE tissues and TMAs with CytAssist. So earlier this year, we added CytAssist to our services, which also has apparent benefit of high coverage of the capture area compared to the direct placement and much higher sensitivity too. So as a case study with CytAssist, we were able to resolve some of the issues that our customers were facing. For example, one of our projects was delayed by 7 months as the customer wasn't able to find a good RIN score glioblastoma tissues. They were working on fresh glioblastoma, surgery tissues, and it was really hard to get a good RIN score sample. And ultimately, we decided to move forward with the project using CytAssist and use the precious samples that they had obtained with low RIN score. Another one of our customers working with diseased and healthy arteries and aged patients had similar RIN challenges besides low tissue amount. So using CytAssist, we avoid tissue optimization, which needs more tissue slides as opposed to just one slide required by CytAssist, and help this customer. In yet another example, we were able to use archival slides from our customer to generate results using CytAssist. So it has really proven very beneficial to add CytAssist to our repertoire. And moving on, as I promised to show the results of our preliminary studies, here is an example of our lung sample characterized using 10x Visium technology. The image here shows the tissue in the capture area and UMI plots and clustering. I would like to further talk about a poster that we recently presented at AACR in April this year. And we used our in-house colorectal cancer and normal samples for this study to provide insights into tumor microenvironment, vis-a-vis heterogeneity and spatial organization of cells. For the sake of time, I am only highlighting a few key results here, but please feel free to follow up with our technical team later today or via email if you are interested in learning more. The figure here shows the heat map, which clearly shows the differential gene expression, the differential upregulation in the tumor below, in the second one. The below tissue is the tumor, and the upper one is the normal. And you can clearly see differential genes upregulated in tumor versus the normal sample. So these violin plots further show upregulation of genes in comparison to the normal tissue. And these upregulated genes also consisted of these 5 well-known CRC biomarkers. Further, this violin plot here shows the upregulation of B-cell associated immune cell biomarkers. And again, we can see we found out that some of the very well-known biomarkers were upregulated in the tumor tissue. So coming back to sample preparation and optimization, let's talk about GeoMx now. So with GeoMx workflow, building a microarray with multiple treatment or tumor samples is cost efficient in terms of reagent consumption. So we can spatially profile multiple tissues on one slide as opposed to multiple runs. And we do offer TMA constructions as shown in these pictures above. And so we can spatially profile multiple tissues on one slide. And as an example, our in-house pathologist recently helped build a TMA with 20 cores. And what they did was they wanted to study breast tissue. And within the same tissue, they wanted to study progression within the normal areas compared to the tumor areas. So our pathologist helped select the cores and build a TMA. Another example is that of our in-house TMA that we use to characterize using GeoMx platform. And I will talk about it in just a few minutes. But before that, I would like to talk about morphology markers, selection and optimization, as well as proteinase K concentration and optimization, which is one of the key steps in GeoMx workflow. So morphology markers are key in the GeoMx workflow as they are used to select ROI and guide further tissue segmentation, as they're specific to the cells of interest in the tissue under examination. And with each different sample or treatment, the concentration of morphology markers and proteinase K needs to be optimized to enable clear visualization of the tissue and segmentation within the tissue. So depending on the tissue, this may take as less as one dry run or more. And here is the breast tumor TMA that we characterized using GeoMx. And this TMA is, we built this TMA at BioChain, and this comprises of 25, these from the second row to the last, 25 breast tumor tissues in duplicate, plus 4 normal breast tissues in duplicate, and 1 placenta tissue for negative control. We used the morphology markers PAN-CK to stain epithelial cells green, and CD45 to stain immune cells red. So for ROI selection, in the whole breast TMA, we selected 88 ROI, that is regions of interest, and each being 400 microns. In the image here, I'm showing you 2 ROIs in particular, B1 on the top and C5 on the bottom. So the left image here shows, the epithelial cells stained green with PAN-CK, and the red area that you see is immune cells stained with CD45. And on the image on the right here, you see blue and yellow colors. Those are the segments that we selected. Blue segment indicates the segment that has only the epithelial cells, which is PAN-CK positive, CD45 negative, while the yellow indicates only the immune cells. So further, we used the data generated from here to create these volcano plots and violin plots. And clearly here in the plot on the left, you can see the differential gene expression between the immune segment versus the epithelial segment. And then the violin plot here is showing the gene expression for only ERBB2 gene in particular. This is the gene that encodes HER2 protein in breast cancer. And, you can see here that this particular gene was upregulated in the epithelial segment in the tumor tissue, tumor cores, compared to normal. So when it comes to spatial workflow, we further have added a few more technologies. We started with NanoString GeoMx DSP and 10x VZM direct placement. Then we added CytAssist and SlideSeq from Curio Bio. And then we added Codex in partnership with Enable Medicine and StereoSeq as well. To summarize, I want to go over some case studies and future prospects. We have successfully been able to process precious customer samples and, generated the data and completed projects with both GeoMx and NanoString platforms. And We have been able to construct TMAs, help the customers with histopathology, and use our in-house FFPE tissues and TMAs to pre-characterize these tissues. And these pre-characterized tissues are also available online for purchase with the section and the data. And we have successfully used archived hardcover slipped slides as well with 10x CytAssist. And also, more recently, we are now getting into further characterizing our pre-characterized 10x pre-characterized oncology samples with analysis such as Codex with the help of our Enable team. So that's something I know Gulpreet mentioned a similar project too. So that's something we are heading into now. And in the coming few months, we are looking at doing more of such projects where we are combining different technologies and, yielding more, generating more results. And we are very excited to further onboard proteomics and StereoSeq technology as well. And to conclude, I just want to highlight a few of our key strengths, which are our biorepository, which comprises of IRB approved rare tissues as well, our flexibility to work with different kinds of tissues, and our experience in tissue handling, TMA construction, and our sample preparation, and our in-house board-certified pathologists, scientists, and the pre-characterized sections that you can purchase online, as well as all these multiple platforms being in one place. So with that, I would really like to thank all of you for your patient listening. If you have any questions, please feel free to ask and reach out our technical team as well.
Hi, everyone. Thank you for joining in today. So if there are any questions that you would like to address, this is a good time to put your questions on the Q&A. So I have a few questions. Can I use spatial gene expression analysis using old FFPE blocks? So I'm assuming this is regarding the 10x Visium, or so specifically if it is for 10x Visium, yes. Now using the CytAssist, you can use archival samples. You can send us either a block or just a slide, and we can move forward with the project. But if you already have a RIN score on these, that would also help us. Currently, using the CytAssist technology, we can use archival samples with low RIN score. I mean, a DV200 score, not RIN, a DV200 scores of around, I think, less than 30%. But if you're looking at frozen, we cannot use archival samples because that is still, we can use archival samples, but if you have some RIN numbers, that also would be of help. So in terms of NanoString, we can actually use any tissues or any slides, and NanoString is pretty robust in terms of FFPE tissue or frozen. We don't have any requirements of RIN or DV200. So another question that we commonly are asked is, can I use, what are the different species that I can use? For example, can I use mouse or rat or other non-human primates for 10x? So we can use, besides human and mouse, currently we have human and mouse panels for 10x on CytAssist. But if you're interested in other types of organisms, such as non-human primates or rat, then we can, primates or rat, we can also use we can use 10x Visium, but we have to do the manual process, which is a little bit more challenging, but I think it still gives excellent results. So that is another common question we ask. In terms of DB200 and Visium for FFPE, I already addressed it. So, because we have the CytAssist, I think that brings down the numbers quite significantly and allows us to use many different types of samples. So another question was, how long does it take for the prep station to process the samples? It really depends on what type of platform. If you're talking about GeoMx, so we don't have to do any early sample optimization. I mean, we have to do optimization, but since we don't have to do DB200 or RIN, that removes that requirement. But then the optimization, depending on what morphology markers you would like to choose, that increases or the time if there is a unique morphology marker that has not been tested by NanoString or in our lab. So that has to be optimized. So really depends for NanoString the type of morphology markers you will be using. If it is something that NanoString has already been used and tested, and we've tested both at NanoString and at BioChain, I think the entire process can be done within 6 weeks. From the time you send us the tissues, we generally do an optimization run that takes around 2 weeks, 2 to 3 weeks, and then do the whole transcriptome analysis, where we can do the ROI selection, the AOI selection, and then the sequencing. But the initial project discussion might take a little bit long, because we have to figure out what type of morphology markers you're interested, number of ROIs, how big the ROI should be, and there's a project design and consultation that might take around 4 to 5 weeks. For 10x, I think if we already have the DB200 or RIN scores, it takes around 3 to 4 weeks for us to complete the process. In terms of analysis, analyzing the data, yes, we can do some analysis, some preliminary analysis, but we have partnered with Enable that can help us do Visium data analysis, and NanoString also is a great resource to do data analysis for us. So these are some of the questions that I have. Are there any other questions? Oh, there's one more that talks about, yes, we have done multiple 10x projects where we do xenografts, mouse to human xenograft. And we are also currently doing a GeoMx project that has mouse to human xenograft. And also the other question that we commonly get asked are using TMAs. How many samples can you put? How big is the actual scanned region? For 10x, it's around 6.5 millimeters. And we can do 2 runs in CytAssist. For NanoString, it's a much larger window. So if you have multiple samples, BioChain has the ability to put them or make or construct a TMA, so that we can actually save some time and money in terms of running. Instead of doing multiple runs, we can make a TMA with around 10 to 20 samples and run it as one run. But we recommend that the tissues all be of the same indication. Like, for example, if you're looking at breast, like the way we did it, then having all breast samples would be more helpful, because we are looking at morphology markers and optimization. So these have been some of the questions. Happy to ask if there are any other questions. Elim, would you like to add something that I've missed?
And no, that's good. So for the DV200, right now we use the sciences, and then the DV200 is about 20%, but it also depends on the, when you have the board, and then the quality of the board and how the fixation and embedding methods or the storage condition or the age of the board, they may affect the quality of the RNA over there. So that's why we will recommend to test the DV200 before we do the sciences. Even the 10x that even is lower than 30%, they can generate the data. However, they cannot guarantee they may be lower than the good RNA qualities. So we still recommend to test the DV200, and also we have archive slides service. If you already put it in the archive slides for maybe several years, we still can do it with the 10x Visium sciences. It's a new technologies, and then we can use the POP methods to remove the cover slip. Even if HNE slinging or the IF slinging, we can still do the Visium, the spatial technology on the archive slides.
Yeah, thank you, Elim, for that. I think that's a good point for everybody to understand that. I think the quality of the block or the tissue really depends on the fixation and the time, how long it's fixed. So we've seen a lot of differences. We've seen very, very old blocks give us great DV200, and if it is fixed well, we've got really good results. But if the fixation and the quality is not great, even newer blocks sometimes doesn't give good quality data, right? So we have come across that. And the quality of the data really depends on how good the block is fixed. So in terms of NanoString, Elim, do you have any best practices in terms of sample preparation, like the quality of the sample?
For the sample preps, and it's also depends on how they they prepare the board, and then how they keep the slides. I mean, if they give us the archive slides, and then how the storage conditions and all these things. So it totally depends on how the customer they storage and the preparation of the slide and the board. So that's why when the samples came to Baoqing, we will do the QC of the samples, and to ensure the win number or the DV200 have been passed the minimum requirement, and then we will get the customer approval to proceed for the sciences.
Okay, so for NanoString, I don't think we require any of these numbers, right?
Yeah, for the NanoString, yes, sorry. For the NanoString, we don't require anything, so we just have to fit in the window for the selection area.
Right. So that's pretty straightforward. So the sample preparation time is very minimal, but what does take time would be the optimization part, which depending on the the morphology markers. But once we do everything, how long does it take for you to identify the ROI? Since we have an internal pathologist, right? Like, how would you recommend a customer be involved in selecting the ROI? Like, how would somebody not staying in the Bay Area, somebody in the either international or East Coast, how would they be able to be involved in the process of selecting an ROI?
Yeah, for the regarding the ROI selections, we have several methods we can communicate with the customer. Normally, if the morphology marker is project specific, and then we will do a dry one. From the dry one image, we can have a meeting with the customer to see what is the region of interest. Is it they will interest in several cells or several morphology, and then they can select the ROI from the dry-run image, and then we can overlay the dry-run image with the real-one image to select the several ROI. On the other hand, we can do a live Zoom meeting with the customer when we have the real-one, and then at the same time, we can have the live meeting with the customer to select several areas. And also the customer can come to Baocheng if they are in the Bay Area, and they're very welcome to select their own ROI in front of the GeoMix machine instrument, and then it's more practical, whenever they can select the region they like or they are interested in. And this is how we communicate and consultate with the customer.
Correct. Yeah. So the three methods: either people can come to our facility and select the ROI, or we can do it through a Zoom, or we can use an H&E and overlay. So the general sample preparation is, let's say somebody sends us, for example, for 10x a block. So we actually section it. We will do a RIN score or a DV200, and then we provide the information. And once that is approved, then we might even do an H&E. We'll do an H&E, and then we will send an image, a Leica image, to our customers, so then they could select the actual region where we would like to actually do the place the slide for 10x Visium. So that is the early part of the sample preparation, where the customer, there's a lot of back and forth between the customer and our team. And as Elam mentioned, for NanoString, there's a lot of back and forth in terms of detecting what type of morphology markers, what type of cells, and then looking at the ROI, selecting the ROI. And we also have an internal pathologist who can help us select these regions of interest. And then the customer is very deeply involved in the process, where we can also help them select. And once that is done, then we go about doing the AOI and selection and the library prep. And then we do the sequencing and provide the data analysis, either with our partner or with NanoString, or we can do some preliminary data analysis. So throughout the whole project, right from consultation, we have both the 10x technical team, our technical team, or the NanoString technical team coming in, helping us assist to design the project, ask the right questions, make sure that the project is being very successful. So are there any other questions that we can address or we did not address? We'd be happy to.
So may I add some information about the data analysis? Regarding the data analysis for the 10x Visium, the 10x provides a free software like the Space Ranger Pi9. This is free, and then they can do some Pi9 analysis, and then after that they generate a C-loop file for the loop browser. They can do the preliminary data analysis in the free software provided by 10x. Regarding the NanoString, the GeoMix instrument, they install the software for data analysis, and then our customer can use the VPN or other methods to access the GeoMix software, and then we can do the preliminary data analysis, or the customer can use the software to do the data analysis. So this is the primary data analysis, and all the data or the presentation generated in the webinars is use this kind of software and methods to generate the preliminary data analysis. For the secondary, we have partners, or you have some customers, they have their bioinformatics team, so it's more more advanced.
So question is, how do we send this data? Because these are really big files, right? So how do we transfer the data or analysis to our customers?
So about the FASTQ and also with the loop browser data, we if the customer have the cow, and then we can upload to the cow. Otherwise, we can transfer with the USB flash drive, or yeah, if they have any faster method, we are happy to communicate.
Mm-hmm. And the same thing with the GeoMix, or is that more part, do they have to access it through the GeoMix machine?
For the GeoMix data, they also only need the FASTQ file. They can generate the DCC. We can also generate the DCC file for them, and then they can do data analysis, either use the bioconductor. This is also a free software online, and if they want to access the GeoMix, we can communicate, and then we can do a further further research on how to use the GeoMix, the software for the data analysis for such project.
Okay. So thank you so much. I think we did address sample preparation, and then project consultation. And as we mentioned briefly that, we do both these technologies, both 10x Visium and NanoString, and data analysis. So since there are no further questions, we would like to say thank you all for joining today, and keep us, we will be presenting more such webinars on our other different technologies. And so stay tuned, and we'll keep sending you more information. Again, thank you so much for joining, and have a wonderful rest of the day. Thank you.